Assalamualaikum and a very good afternoon. So I pre-recorded this lecture first in case we want to save some time later on. Okay. So for the next topic is going to be about the fish, not meat and fish. This is basically focusing on the fish only. The meat will be taught by Professor Nazani. But basically they have a relationship with each other because both of them contains muscle protein. Okay? And the structure is the same, just a little bit of different in terms of the color of the meat, the structure and so on. But the building blocks, they're supposed to be similar. I'm actually not an expert, or I never teach about meat and fish before. My forte is the human biochemistry. So this is just a summary from the textbook that I told you earlier, right? Okay, for the introduction, we take a look at the fish muscle first. So basically, fish muscle is a layered rather than bundle muscle, and it is much less than other type of meat. That's why it is softer. That's why when you cook the fish, it is easier to get tender compared to the normal meat. Each segment or sheet is called myomere or myotomes and separated by connective tissue, known as myocomata. Salmon is easy to study due to the distinct separation of these segments. Okay. For example, as you can see from this figure or picture, the myocomata is the white lines that the connective tissue that separated the fish muscle, and this pink color is the myomere or the sheet of the fish. Fish can have red, pink or white muscles depending on the fish. The color of the red because of the presence of the red pigment such as hemoglobin. White because the lack of pigment, often utilized as explosive and anaerobic energy. What it means that if the fish is red, usually the fish is very active. But if the fish is white, the meat means that the fish commonly use anaerobic respiration, means that they are not very active. And they can swim very fast, or when you caught them on the fishing net, they can move very violently, but for a very short time, because anaerobic respiration is less efficient than the aerobic respiration. Okay, so if you have aerobic respiration, you will need a lot of hemoglobin to bring the oxygen into the muscle. And some fish can demonstrate pink color muscle because they get this pigment from the carotenoids from their food. Okay, for example, the salmon. Okay, for that one is for the fish, and the subsequent slides is going to be about fish and other kind of seafood, for example, crab, mollusks, and so on. So many things can affect the freshness quality of fish, including processing parameters and different stages of post-mortem transformation. Post-mortem means that after the fish has died. Perishability of food is fast and can be affected by following factors. The first one is biochemical changes of glycogen. Second one is the nitrogenous compounds formation due to the protein degradation. The third one is because of the rancidity of the lipid of the fish muscle. The fourth one is the changes in pigments or discoloration. And the last one is physiochemical changes, for example, reduced pH because of the formation of the lactic acid, collagen degradation, nucleotide catabolism. This one is nucleotide, nucleotide. And the final one is free fatty acid accumulation, because some fish, they contains a lot of fat. So when they die, the fat will undergo lipolysis, being broken down by the enzyme and form the free fatty acid. So sometimes when we want to know whether the fish already died for a long time, we can measure the amount of the free fatty acid of the fish, okay? Or we can use other parameters, for example, the residivity of lipids, the formation of the nitrogenous compound, or the reduction of the glycogen inside the fish. Right? Three types of processing will be focused on. The first one is freezing, and the second one is drying, and the last one is heating. Okay, for the first one, which is the glycogen degradation, glycogen is a highly branched glucose molecule, very similar to starch. Okay, but the breakdown of the glycogen is a little bit different from starch, right? And it can act as energy reserve after you finish off your ATP. This one is usually similar between human and other animals. Okay, for example, fish and other seafoods. When seafoods are caught, they will drain their glycogen reserve because they struggle a lot and they use a lot of energy, and it will trigger asphyxia. I'm not sure how to pronounce this one, but basically they don't have enough oxygen. And it will result in the formation of many byproducts, for example, formaldehyde, ammonia, which give the unpleasant odor to the fish, inorganic phosphate, ribose phosphate. It can produce lactic acid because of the anaerobic respiration from the energy formation during the movement of the fish. And this reduce in pH because of the lactic acid can cause contracting of the tissue and induce rigor mortis, post-mortem changes due to the stiffening of the body muscles. So this is the example of the structure of glycogen. You can see it's a very highly branched glucose molecule, and in order to break down this into usable glucose, you need two types of enzyme. Okay, which is the first one is alpha-1,6 glycosidic bond enzyme, and the second one is alpha-1,4 glycosidic bond. Okay, because this one is the branch, we call it the branch point is alpha-1,6, but for the non-branch point, we need to use alpha-1,4 to break down the glucose from the glycogen molecule. Okay, there are three types of seafood proteins similar to other muscle food, and they can be classified as sarcoplasmic in 30%, roughly 30% of the total fish muscle. The second one is myofibrillar, 40 to 60%, and finally stromal or collagenous protein. The sarcoplasmic muscle usually consisted of myoglobins, enzymes, and albumins. Myoglobins is one of the red pigment that can give color to the fish. So if they are degraded, the red color of fish can turn into something else. Okay, and we know that the fish is not very fresh anymore. The enzymes that is present in the seafood, they can cause quality deterioration after death and before bacterial spoilage, because if the fish died for too long already, the bacteria start to grow, then the spoilage of bacteria can cause other types of degradation of the fish muscle and also protein. So myofibrillar usually consists of myosin type of muscle, actin, tropomyosin, and troponin C, troponin I, and troponin T. The changes during rigor mortis, myofibrillar can change because of rigor mortis, because of resolution of rigor mortis and long-term frozen storage. So remember, not only the death can cause the degradation of the myofibrillar, also if you keep the fish for too long in the freezer, it can also cause changes. The degradation of myofibrillar can also lead to the loss of integrity and gelation, so there is no firm texture and mushy when you cook the seafood. That's why some people, they will press or push the seafood to know whether it is fresh or not. Okay? Because the one that is not fresh, they are very mushy and the substrate is not very springy anymore. The degradation can also lead to the loss of functional characteristics, namely solubility, water retention, gelling ability, and lipid emulsifying properties. And it may get worse when proteins are cross-linked due to the presence of formaldehyde. Okay? And it can lead to the tough, chewy, stringy, and fibrous seafood. Also, the third one, the stromal, mainly consisted of collagen and elastin from the connective tissue. And the degradation of stromal protein can lead to honeycombing in skipjack tuna. I'm not sure what is this, but basically it will form like a honeycomb structure on top of the flesh, and mushiness in freshwater prawn. Okay, because of the degradation of collagen. Probably because prawn contain a lot of collagen. Okay, so for the biochemical changes in non-protein nitrogenous compound, white meat generally contains less non-protein nitrogenous compounds. Okay, there are protein nitrogenous compound, which is the 21 amino acid, and the non-protein the protein that is not within the 21 or 20 amino acids. Okay, the essential one. So white meat contains less than the dark meat. For example, fish contains up to 15% of NPN compounds. Clupids, I'm not sure what type of seafood is this, can contain up to 18%. Crustacean up to 50%. And certain big fish, for example sharks, contain up to 55% NPN compounds. Okay? Probably because it contains lots of protein or lots of muscle. Okay, the degradation of the NPN compound can lead to the formation of many chemicals, for example, imidazole dipeptides, TMAO, and its degradation product, for example, urea, guanidine compounds, nucleotides, and DMA. Okay, you will see a lot of TMAO and DMA after this. This can also, the degradation of this compound can also produce odor. Okay, for example, from the ammonia production from urine and ATP, formaldehyde from TMAO, which can cause certain food safety issues. For example, poisoning, stomach ache, and so on. Especially in shark. So not like we eat a lot of shark, but if the shark is left for quite some time after death, it can develop a lot of this ammonia and also the TMAO. However, if you still eat shark, it can be avoided by bleeding or wash the shark right after you catch the shark, right? Because you wash away probably most of the precursors that can lead to the formation of these compounds. The breakdown of nucleotides can be positive or negative. For example, inosine monophosphate can be desirable flavor in dried fish. If you notice certain dried fish, the fermented fish, they have a very strong smell. It can be off-putting for some people, but usually this is the characteristic that people want in the fermented seafood. Okay? And this is caused by the breakdown of nucleotides that produce something else. That give the odor and also the flavor to the dried fish. Okay, I will continue in the second lecture. Okay, because the limit is 15 minutes. Thank you.